All right. So, as was mentioned, we have for several years as a society been doing some work with the dinosaur tissue that has actually been found by a lot of different labs. Again, for those that maybe weren't here last evening, the Creation Research Society is a society founded back in 1963 by a group of scientists who are also biblical creationists as a society of professionals supporting the biblical teachings of creation, the biblical teachings of origin. There is our website, creationresearch.org. You can contact us through the website. Anybody can be a member of the society. It's international. We have a number of Canadians also that are members. Anybody can be a member of society if they agree with our statement of faith, our position statement. And then anybody who has an advanced degree in science can also become a voting member. And because this represents so much of the last several months or years even of my life, this is our brand new Andel Research Center and also the Society's Operations Center. This is our lobby area in the picture, and of course on the outside. Just want to brag about that because we're really excited about the opportunities this new facility poses to us. Not only the location of it, but just everything is now modern. We have a brand new, very large laboratory in the back where we hope to do a lot of research once we really get it set up and going. So this brings us then kind of to the question at hand, and we're going to start with Charles Lyell. Back in the 1830s, Charles Lyell wrote a series of books called Principles of Geology. Now you have to recognize Charles Lyell did not have any formal geological training. He was a lawyer. But he used his, even as Stephen Jay Gould acknowledged, he used his lawyer arguing skill to basically use words as support of his position rather than evidence. Lyell argued that all geologic change is slow. It takes a long time to form a mountain. It takes a long time to form a canyon. It takes a long time for those layers to form. And so because all geologic change is slow, the earth has to be very old, and it takes a long time for the earth then to present, if you will, what we see today geologically. That's a result of millions upon millions of years of geologic activity. Darwin even recognized the significant impact that Lyell's writings had on him. He said that I can't express enough that half of my ideas come out of Lyell's brain. So Darwin's thinking of slow biological change is a direct product of Lyell's thinking of slow geologic change. But now if we kind of dig in a little deeper, we begin to recognize that Lyell himself admits a lot of his motivation for making the arguments he made was not because of the science. It was because he was an atheist, and he wanted to, as he said it, free the science from Moses. In that respect, the science being geology. Because at that time, even though maybe not everyone that was in geology at that time would have said the earth is just a few thousand years old, they still by and large were dominated by the idea that there had been a great flood. And the Great Flood is what accounted for a lot of the geology we see today. And so Lyell, by trying to make his arguments, his thinking was, I want to get all that out. I want to remove all that so that creation and the flood and all that has nothing to do with geology. And that was, as he acknowledged, really a lot of the motivation behind what he wrote and why he wrote what he wrote. Okay, so that brings us to today. And what I would view then as a standard evolutionary interpretation of the fossil record. We would see in some fashion a fossil record presented in the geologic layers, and evolutionists would interpret then that, okay, so this one transformed into this one, which evolved into this one, which evolved into this one. And so they would argue that the geologic record sets up evidence of biological transformation that's occurred over great ages of time. And that would be their interpretation of the fossil record. Okay, the creationist interpretation is, no, we argue that the fossil record as a whole, or as a conglomerate, if you will, was laid down mostly by the flood. Not all, but mostly by the flood. Therefore, the fossils in the fossil record represent animals that were not evolving into each other over tens of millions of years, but actually were contemporary. So shown there, the fish and the land creatures, all we find in the fossil record, were contemporaries of each other. They were all buried by the flood. So two totally different interpretations. One requires massive amounts of time. The other doesn't require any time at all, except the time for the flood itself. So when we're getting then into history of the Earth and the age of the Earth, I would challenge what we're looking at is if everything takes millions of years, then you're looking at Lyell's timeline. But do you fit Lyell's timeline into the Bible timeline? And of course, some people have tried to, but I think unsuccessfully. So what we really have is a Bible timeline versus Lyell's timeline, which is correct. Okay, so if we look at the standard geologic interpretation, they're going to say that dinosaurs lived during the Cretaceous, the Jurassic, and the Triassic periods of Earth history, and that these date back from 65 million years to some 250 million years. And dinosaurs lived then in that period of time. Interestingly, in the movie Jurassic Park, it was pointed out to the writer, Michael Crichton, that all the dinosaurs he featured in the movie were actually Cretaceous dinosaurs, not Jurassic dinosaurs. Just always kind of amusing because people think of Jurassic as dinosaur, but Cretaceous is really the period of the dinosaurs we're thinking of when we look at our textbooks and such, as far as T-Rex and Triceratops and such. So again, recognizing dinosaurs in an evolutionary standard geologic interpretation lived over many, many millions of years throughout 3 entire geologic periods. All right, so this brings us to then an understanding of what bone is and how bone fossilizes. Bone is, you start bone by laying down a matrix of a protein called collagen. And then you have various bone cells that function it is to take calcium and phosphorus and coat that collagen and make a mineral coat of that matrix. Bone is really fairly porous, some bone more porous than others, but we have to recognize that bone is not a rock. Bone is living tissue, even if it is highly mineralized in some areas. So we got to recognize what bone is. So forming a fossil is a process of, in essence, making the bone into a rock of a fashion anyway. So let's say we have an animal that dies and its bone gets buried. Okay, that introduces then groundwater into the bone, and the groundwater will bring with it various components such as silica, a lot of silica, also calcium carbonate and such. And the groundwater will bring in this outside material. It will wash out the calcium and the phosphorus of the bone, and there'll be a replacement. So when you're done with this, now you have a fossil that does not contain the biologically active levels of mineral. It now contains a different composition of mineral depending upon the surrounding mineral levels. You know, how much iron, how much carbonate, how much silica is in, or sulfur and such is in the fossil depends on what's in the surrounding area that the fossil was buried. And so the question I asked then is just how long does this take for this fossilization process to occur? Because apparently from the data that we're now being presented with, it must take some 200 million years for this fossilization process to occur. This brings us to Mary Schweitzer's work in 2005 and then some follow-up papers in 2007. Now, to lay down the background for a moment, we have to recognize that Mary Schweitzer's 2005 paper was not the first paper that reported the possibility that there was some kind of biomaterial still left in dinosaur fossils. We have papers dating back to the 60s that are speculating that it looks like there's some amino acids here. It looks like there's maybe some fragments of proteins in this fossil. They were typically just, you know, ignored, typically just hand waved away because, well, that's just contamination. That's just amino acids off of your skin. That's just amino acids from bacteria that have gotten in. So by and large, it was assumed that it's contamination. We don't need to worry about it. And then when Schweitzer's paper came out, first off, she published in the journal Science, and the journal Science is the second largest scientific publication in the world. Second off, she published in color, and I don't think it should ever be minimized, the impact of color. And so this really caught the attention of the scientific community, unlike any paper on this topic had ever before. And the methods she used were more sophisticated than methods that been used before, so it was a little more difficult to just dismiss it as contamination. Since 2005, there's been a number of studies that have been done by a number of different labs on a number of different fossils in a number of different countries. I have to emphasize that this is not just all the product of one fossil in one lab in North Carolina. Okay, a lot of different labs, a lot of different researchers using a lot of different fossils have found tissue of some fashion. So what we have that we can summarize is the pliable tissue has been found in a variety of dinosaur fossils. Morphologically distinct cells are present in some of this tissue that's been isolated. Collagen fragments are frequently detected, but other proteins such as actin, myosin, and tubulin are also frequently detected as well. It's not just collagen. So if we step back and say, okay, there's biomaterial still in these dinosaur fossils, which are supposed to be at least 65 million years of age, then we have a problem, because the presence of this tissue is a direct challenge then to the standard assigned dates, because you don't have tissue lasting that long. At least that would be certainly what we would understand as biologists. Tissue doesn't last 60, 70, 80 million years buried in the ground. So this is when the Creation Research Society started its iDino project. Stands for Investigation of Dinosaur Intact Natural Osteotissue. And yes, we wanted it to stand for iDino. Okay. We felt that it was not only appropriate for us to get into this work, but we also felt it was necessary because, in all honesty, we were a little bit fearful that the evolutionist community may not do some of the work that we feel needs to be done, and therefore will do it if they're not going to do it. So that was another one of our motivations of getting into this field. Okay, in 2012, the Creation Research Society sent a team to a formation, to an area in eastern Montana, in the geologic area of the Hell Creek Formation, and we found a Triceratops horn. And this is the Triceratops horn we found. Its total length in the ground was 46.5 inches. Now, it was obvious that that wasn't the full length of the horn. So this horn, as when it was on the Triceratops himself, was actually probably closer to 50 inches. So it was a monster-sized horn. So this Triceratops would have been kind of, you know, the great-great-grandpa of the herd. It was also heavily encased in sandstone, and it took a lot of chopping and digging and pounding to loosen that horn to take it. Now, since then, it's been a little amusing to me that this particular picture has made its rounds all across the internet. You can find all kinds of websites, including websites about UFO invasions and such. So I, was not aware that we were also finding aliens, I guess. But anyway, that happens to be my arm. And so somehow I need to copyright my arm so you can't use my picture of my arm on your website. But I just found it several years ago that suddenly it was all over the internet. And just all kinds of websites have this picture, even if the website has nothing to do with the studying of dinosaur tissue. So from the horn that we excavated, here is a picture of some of the material that was extracted. And this is on the cover of the American Laboratory magazine. And you'll notice it has a very tissue texture characteristic. Also, those dark spots in there, those are still intact osteocytes, still intact bone cells. So those are dinosaur cells that you see still present in that tissue. If we look at some of those cells even higher magnified in electron microscopy, we see they have the very distinct characteristic of vertebra osteocytes. That's how we know they're not contamination, because contaminating cells wouldn't look like this. These are very distinct bone cells. And you notice also what the arrow is pointing at in the one slide, or in the one picture, those are Philopodia. Now, Philopodia are extremely narrow, very fragile extensions of proteins. And yet look how they're still intact. They're so carefully preserved. So this tells us the very wonderful level of preservation that's occurred in these fossils, which is allows us to find this material, what allows us to find the tissue. And this presents then a situation of how could you have such pristine preservation in a very non-pristine condition, a very non-pristine fossil. In fact, when we first excavated the horn and we pulled it out of the ground, and I was sitting in the back of the pickup truck with the horn to stabilize it, it was already broken in pieces because we didn't take the time to bring the whole horn out of one piece. And the museum people that were our guides, they were just beside themselves because this was the biggest horn they'd ever seen. And of course, they, as far as they were concerned, this would have made a beautiful museum display, but we were taking it to do scientific study. You know, it has to be sacrificed for scientific study, I guess. So we were going to break it up and destroy it. And so I was sitting back there with the horn, stabilizing the pieces on the pallet that we had carried it out on. And I was noticing how wet and muddy it was. And I thought, gosh, I'm a younger Earth biblical creationist, and even I don't believe there's going to be anything in this fossil. And sure enough, there was. There was still a lot of tissue. And so it's difficult enough to explain how that tissue could have survived a few thousand years under such a low preservation condition, but how would you explain it surviving some nearly 70 million years? That, I think, now you're borderlining on the, you know, absolutely irrational Alice in Wonderland stuff. Here is a second specimen that we work with. This is a vertebra of a Thescelosaurus. Okay, now if you'll notice the end vertebra, the vertebra on the very end, it was so damaged that the museum didn't want it. So they simply wrapped it up, put it on a shelf, and didn't think anything of it. Okay, a few years ago, I then had a young lady who wanted to serve as an intern in my laboratory that came from the museum and brought that particular vertebra with her. And she was studying under the microscope and kind of just digging around and taking some tools and scraping it off and such. And lo and behold, this is what she found. And then when we took that piece and some other pieces too, and we washed it off some, and then looked at it as an isolated piece, see, it looked like you have a little piece of dinosaur steak there. It has all the characteristics of the type of tissue you would expect that would be biological, very stretchy, has a certain consistency, a certain thickness to it that is consistent with vertebrate tissue. So what we have is we have some preserved dinosaur tissue still in that vertebra from that Thescelosaurus. Other studies then, not ours, but other labs have done some studies where they isolated some blood vessels from a dinosaur fossil. Now think about that. They pulled out and extracted blood vessels. The blood vessels still had the characteristic translucency, the characteristic elasticity of an actual living blood vessel. So it still retained a lot of its biological characteristic. And in the blood vessels, they found fragments of collagen, tropomyosin, actin, histones, tubulin, myosin. In other words, all of the proteins represented that you would expect to find in blood vessel tissue of a vertebrate animal. So this leads to then a conclusion that when we take into account the studies that have been done over the years of collagen degradation, there is no study done that has demonstrated that collagen will last any longer than 4 million years, actually less than 4 million years. And in fact, that's being very, very generous. Very generous. I threw that in there so there wouldn't be any exception that somebody, what about this study? What about that study? There is no study that does anything up to 4 million years as the length of time that collagen would be able to last. And notice also, see, this is under ideal conditions. And I guarantee that a fossil buried in the ground is anything but ideal condition. So how do you have this biological material still present after such a length of time when there's no evidence, there's no experimental verification that these proteins could last that long? Collagen is by far the toughest of the bone proteins, but see, that's not the only protein that's been detected. Everybody focuses on collagen, but remember, they've also detected actin and myosin and those other proteins I just mentioned. They don't have near the stability that collagen does, so they'd only last a fraction of the time. So even if you argue that collagen can somehow make 4 million or 5 million or 10 million years, actin and myosin and tropomyosin and such, they don't. So their presence then indicates also even less time that you would expect that you would be able to find collagen. So the time frame, in other words, the ceiling that you have of how old these fossils could be is far less, far less than 68 million years. Now, one of the problems faced with saying that the fossil is tens of millions of years old, but it still has collagen, pieces of collagen in it, is that collagen is most stable when it's at its most stable, which would be what you'd need for it to be able to survive so long. When it's at its most stable, it's also at its most temperature sensitive. And this was pointed out in an article in Science 2008, where they were arguing that Schweitzer's work cannot possibly be correct because the Hell Creek Formation, at the time this dinosaur was supposed to have been buried, in other words, using their standard calculations, their standard geologic interpretation, at the time this fossil was supposed to have been buried, Hell Creek would have been a megathermal environment. So in other words, the collagen had to be its most stable to last so long, and yet that would have made its most sensitive at a time when the Hell Creek was its hottest. And so they argue that in this environment in Hell Creek, you would only have a half-life for collagen of 2,000 years. And obviously, if you half-life 2,000 years, that does not add up to a survival rate of 68 million years. So there is a clear problem in the biochemistry versus the standard geologic dating. Now, this brings in a situation of just how reliable are the protein decay curves. In other words, these studies done to determine the degradation rates of proteins, are they really reliable, or are there some problems to them? Okay. The Creation Research Society published a special report in our Creation Research Society Quarterly, which is our publication. We publish it 4 times a year. Okay, we published a special report in 2015 of our findings up to that point. Okay, Fazell Rana, who works for the Reasons to Believe Ministry, which while they identify themselves as creationists, they certainly don't agree with our interpretation of the age of the earth. And Fazell Rana then wrote a book called Dinosaur Blood in the Age of the Earth. And in that, he very heavily referred to our special report, which we appreciate, thank you. Okay. And one of the things he argued was that you cannot apply the high temperatures used in the basic biochemistry studies, the thermokinetic studies, to explain then the collagen decay rates in fossils over millions of years. And his reasoning went that you cannot extrapolate that much, although biochemists do it all the time. And I challenged him then in an article I wrote for Answers in Genesis, I challenged him then to provide some experimental verification of his assumptions. I said, you can assume all you want, but give me some experiments that actually back up your presumption. So interestingly enough, he took me up on that. And I got 2 articles. I really felt honored. I got 2 articles as a rebuttal. And in that, he tried to claim then that you couldn't use these experiments because the temperatures used in the experiments create a misinterpretation. Let me explain. He cited a 1972 study that shows that collagen's denaturation temperature is way below the temperatures typically used for these thermokinetic studies. Thermokinetic studies often use temperatures of 80, 85, or 90 degrees centigrade, very hot. The reason for that ultimately is simply because when you have a protein as stable as collagen, if you just set it on the kitchen table and waited for it to decay, it'd take years. And of course, your graduate student likes to graduate at some point in time, and you like to publish at some point in time. So you're not going to wait 10 or 15 years for you to get the results. So you speed up the results by putting it at higher temperatures. It's all well established. You can read it in biochemistry books. Biochemists have no problem doing this at all. It's been done for, thermokinetic studies have been done for decades. What Dr. Rana was trying to distinguish was, he's saying that using the high temperatures, you're going to denature. Denature is you're breaking up the 3-dimensional structure, breaking up the quaternary and tertiary structure of the protein down to its basic primary structure, which is really just the DNA sequence. And then the degradation is the breaking of the peptide bonds disrupting then the structure of the primary sequence of the protein. And his argument was that because the high temperatures are going to denature so quickly, now it makes the protein much more vulnerable to degrading, which wouldn't happen that way in nature, wouldn't happen that way in a natural setting. Okay, so he assumed because it wouldn't happen that way, then the mathematical conversions that are done, extrapolating from a 90-degree degradation to a 10-degree centigrade degradation, is what would be the temperature typically for the fossil. That means the mathematical conversions are incorrect. And so you can't draw conclusions from these previous studies that have been done. However, he made several biochemistry mistakes in his arguments. One of the most basic one is that the 1972 study that he cited uses muscle collagen. And so the denaturation temperature that they pointed to is the denaturation temperature of muscle collagen. But the thermokinetic studies that I and other creationists have cited use bone collagen, and bone collagen denaturation temperature is close to 90 degrees. It's very hot, much higher than muscle collagen. So any conclusion you draw from muscle collagen does not apply to bone collagen. So he made a fundamental mistake as far as wrong type of collagen. So the bone collagen being much more stable, much higher denaturation temperature is much more amenable to correct calculation using high temperature extrapolating to low temperature. Okay, so another laboratory has challenged, okay, we're not questioning the thermokinetic studies because this laboratory does thermokinetic studies all the time, but we're challenging that we think it's contamination. And their argument for contamination was that for the most part, when the sequence is being analyzed of the collagen, it's typically just one sequence, and that sequence correlates to ostrich collagen. So we think that perhaps there's some contamination of ostrich collagen in the mass spec, which would be really pretty bad news for the analytical lab that they have that much of a problem of contamination problem. But they're arguing that because it's always the same sequence and it lines up with ostrich collagen, there somehow is this continual contamination of this one sequence. Now, I don't know if it's in direct response or it just happens to be coincidental, but not long after that, a group of researchers looked at and investigated more collagen sequences. In other words, they dug deeper, if you will, and they found 6 additional collagen sequences. And in fact, these collagen sequences correlate closer to crocodile collagen than to ostrich collagen. So essentially, as a rebuttal, it's not just one sequence of collagen that's found, it's a number of sequences that's found. So it's not just easily dismissed as it's just the repeated contamination of this one type of collagen. But also to address that, we don't have to do the extraction techniques to detect collagen. We can use a process called Fourier transform infrared spectroscopy. This is a non-invasive technique. You're simply using particular energy wavelengths, energy of molecules within the collagen to detect then the presence of collagen. And so, for example, a theropod rib that was dated supposedly 195 million years of age was reported by using the FTIR analysis to contain type 1 collagen. And then another analysis of a crustaceous bird, in other words, a bird supposedly living at the same time as several of these dinosaurs. Again, they used FTIR analysis to detect then collagen as well in the bird fossils. So we have a non-invasive way of detecting collagen that would not lend itself to potential contamination by extraction and analysis techniques. Also, we can use second harmonic generation imaging. And this was a slide supplied to me by one of my colleagues, Brian Thomas, who works at the Institute of Creation Research. And he took a Triceratops femur and he analyzed it under second harmonic generation imaging. And what he found then, when you actually analyze for collagen, you see is the red on there, that's collagen. So we got little spots of collagen scattered, little pockets of collagen scattered throughout this fossil. Again, non-invasive, you're not creating a contamination by extraction, you're not creating contamination by having something residual in your analytical equipment. So we have Two, and there's a number of them, I'm just taking the time to look at two additional methods besides mass spec, besides the typical way of sequencing and analyzing collagen, that are verifying the presence of collagen. And contamination is not going to explain the presence of collagen for all of these different techniques. Okay, so this brings us then to a comment made back in 2015, where they suggest that the preservation of this soft tissue is common preservation. So that's even in 2015, how frequent the detection of this type of biomaterial had become. And since then, obviously, it's become even more common to find biomaterial in so-called ancient fossils, and not just dinosaur fossils. Supposedly in a 500 billion year old beard worm, they have found fragments of proteins. You know, that's 500 million years and still got fragments of proteins. Still have evidence of biological material in very ancient, by their time frame, very ancient fossils. So it's not just dinosaur. So this brings us to a statement that Mary Schweitzer herself made in 2014 in an interview. She said, that leaves us with two alternatives for interpretation. Either the dinosaurs aren't as old as we think they are, or maybe we don't know exactly how these things get preserved. Fair enough. I think that really kind of summarizes where we are as scientists in investigating this. But here's the problem with what she said. Dinosaurs aren't as old as we think they are. That's not even on the table. You're not even allowed to discuss that. Of course they're old. It can't be that. So it has to be, there's a preservation technique that we just don't understand. There's some kind of preservation going on that we don't have any biochemical understanding of. And that's the problem I have, is they have closed off one of the possible alternatives simply because of their paradigm, because of their indoctrination. No, you can't discuss the age of the dinosaur. That's already established. Well, it's not established at all, but it's not allowed to be talked about. So here we have, in essence, a type of a cancel culture in science. All right, so that brings us then, okay, if we're going to say you're preserving this biological material for this longer period, then how does it survive? How do you preserve for 65, 75, 85, 150, 200, 500 million years? Okay, and this brings us, of course, to the most popular of all the models, and that's what I'll just simply call the iron model. And that is that iron will stabilize the tissue. Blood contains iron, which is in hemoglobin. Blood has a very high concentration of hemoglobin, therefore it's a high concentration of iron. And so the blood will preserve the biomaterial. That is the synopsis, if you will. So let's break this down for just a second. Okay. First off, iron preservation involves what's called Fitton chemistry. Fitton chemistry is iron will catalyze reactions within biological material that will release ions and free radicals. The ions and free radicals, especially the free radicals, will then react with other molecules, such as proteins, causing them to cross-link. So, for example, if we just do a very simple demonstration, okay, here we have the free radicals reacting with some of the amino acids on the protein, and it's causing it then to cross-link together, and essentially like taking a string and knotting it up. The more knotted up it is, then the more resistant the protein is to enzymatic and microbial degradation. And this is true. I mean, certainly no one's going to challenge the chemistry of this. It will increase its resistance. But here is the situation. Does it increase the resistance to the point of saying that now we can take biological material that would ordinarily only last a few thousand, or maybe at most a few tens of thousands of years, and suddenly last for tens of millions of years? Okay. So as experimental verification of her model, Dr. Schweitzer and her team did a study with ostrich blood vessels. And what they did was they soaked the blood vessels in hemoglobin and they soaked them in water. And then they examined the blood vessels and determined then that after 3 days, the water-soaked blood vessels had a high level of decomposition, where after 2 years, the hemoglobin-soaked blood vessels had very little decomposition. They were mostly intact. So because the difference between 3 days and 2 years is 240-fold, they say, see, 240-fold increase in stability. So if you argue that collagen can easily survive for 1 million years, or will survive for 1 million years, 240 times that, so you get 240 million years survival. And so that then became the popular explanation. It's been offered time and time again. Here's the solution. Iron does the job. Iron's the secret. We're done. Okay, just to use the pictures that she published, we would see here that the hemoglobin-soaked blood vessels versus the water-soaked blood vessels. Now, the first thing you have to remember is the water will be a leaching element. It will wash out all of the pigmented chemistry in the blood vessel. So the blood vessel will just visually appear to be very anemic, very pale, very thin, whereas the hemoglobin soaks, that's introducing a lot of hemoglobin, which will be very bright red. And so it just simply by visually just looks more healthy. When you go to a grocery store and you have a choice between a brown package of hamburger and a red package of hamburger, virtually everybody takes the red package, even though there probably is not any difference other than the hemoglobin is more reacted in the red than it is in the brown. But as far as the quality of the meat, it's going to be the same, almost certainly. So we have that natural tendency to think that the bright red means better preservation. And I'm going to suggest that even scientists aren't immune to that. I think that the bright red and that nice, beautiful color certainly gave the impression that the red blood vessels, or excuse me, that the blood vessels themselves were more preserved. Now, I'm not going to discount 2 years. Certainly 2 years is not trivial, but you have to recognize you're taking 2 years and extrapolating it to 70 million years. And the 2 years is at a constant temperature, where the 70 million years is at a fluctuating temperature. If you understand biochemistry, you recognize that one of the fastest ways to accelerate the degradation of protein, or any biological material, but protein specifically, is to fluctuate the temperature, to warm it up, cool it down, warm it up, cool it down. If you recall with the Triceratops horn that we pulled out of the ground, it was buried in sandstone, but it wasn't buried that deep. It was a little over a foot down. That meant that it still was subject to the changing temperatures that eastern Montana experiences. Eastern Montana can get fairly warm in the summer and get very cold in the winter. And what you have is this constant fluctuating temperature that works to break down biological material. And so if you're going to say it's been experiencing that for millions upon millions of years, see, that just the fluctuation itself would dramatically accelerate the degradation of the biological material. So again, when you're extrapolating 2 years as a constant temperature to 70 million years at fluctuating temperature, see the application, if you will, begins to break down. Also, in the study, they admitted that they got insufficient release of the iron, that is the hemoglobin in the red blood cells, naturally. In other words, the red blood cells didn't quickly break down. And therefore, they weren't getting enough iron released into the solution to be able to enter into the blood vessels themselves. So they had to mechanically disrupt the red blood cells to get the hemoglobin to release so that the hemoglobin would release the iron. So by their own admission, they're actually suggesting that this approach will not naturally work, certainly under the condition they were using. Also, you have to remember that most dinosaur fossils, and particularly a lot of the ones that have been studied for biological material, aren't found as a full skeleton or aren't found buried on top of each other. They're what we'd consider highly disarticulated. In other words, they're scattered all over the place. So they're not buried in a bloodbath. They're not sitting there with just in a tank of blood soaking up all of the hemoglobin to get plenty of iron. They're widely scattered. There would be a minimum amount of blood and therefore a minimum amount of iron. We also have to recognize that cross-linking reduces elasticity. The more cross-linking you'll have, then the greater the preservation, but the less elastic the tissue will be. But remember, this elastic is still this tissue is still very elastic. You saw the slides, the pictures I was showing, and that's not just mine. That is most of the tissue that labs are extracting from these fossils is still highly elastic. That's part of how they know it's the tissue, is because of its elasticity, because fossils aren't elastic. And yet, if you have the cross-linking necessary to impose that long a preservation, they should be like a hard piece of plastic. They should be very non-elastic. In fact, that's one of the reasons the plastic is not very stretchable, because, or at least the hard piece of plastic is not very stretchable, because they're highly cross-linked, which makes them last longer, which is why people complain about being in landfills and such, because they last so long. but they are not going to be elastic, but yet the tissue being extracted is highly elastic. So it doesn't have the cross-linking that would be necessary to give the preservation that this model is purporting to give. And also, and this I think is not trivial in any way. In fact, it's probably my single biggest criticism of her work. Up until that time, Mary Schweitzer has done some, and after this study, she's done some excellent work, very careful and very meticulous in the analytics that they've done. And yet in this study, they got very sloppy, in my opinion. The only measurement they did of the blood vessels was strictly visual. They looked at our slides and, oh, this is more degraded than this. They didn't provide any protocol of how they measured degradation, like we're measuring the cell wall, the vessel wall thickness, we're measuring the length of the vessels. They didn't provide any protocol for anything like that. It was just strictly visual. You know, oh, this looks more degraded than this. And if you don't have any analytical or objective protocol, it becomes very, very subjective. And then when you're trying to then impose a subjective interpretation, and as we recognize, that's what it is, a subjective interpretation. Now, it may be that you claim, but everybody sees it's obvious. Perhaps so, but on the other hand, there's a reason why science likes to use objective interpretation or objective protocols or objective analysis, because you don't have that subjectivity entering in that's maybe fudging the data a little bit, maybe kind of skewing it some. And so the fact that they didn't have any kind of protocol that objectively measured the degradation of the vessels, I think is a huge negative to this study. But then it's also been pointed out, not only by creationists, but as I show you the publications, by other evolutionists, that there simply is not enough iron in dinosaur blood. In other words, in a vertebrate creature, such as a dinosaur, their hemoglobin level isn't going to be satisfactory to contain enough iron to be able to do what this model purports it to do. So it's been suggested that we have to bring iron in from the exterior. We got to bring geologic iron in. Now, there are several problems with geologic iron, but one of the problems is it's highly insoluble. Highly insoluble. Now, you might solubilize it some with certain microbes, but then you got the microbes present, and they're messing up the tissue. So you kind of got pick your poison. Do you want microbes to solubilize the iron, in which case they're also probably creating an environment that is solubilizing the tissue as well? Or do you go with the very insoluble iron? And of course, insoluble geologic iron requires a very low pH for it to become soluble. And that low pH itself can be very damaging to the fossil, excuse me, to the tissue in the fossil. So you kind of have a situation of counter purposes. The conditions to have enough iron to preserve the tissue are also going to be conditions that will degrade the tissue. So which is happening first? It's a race. Can we preserve before we degrade? Because of the inadequacy or the incompleteness of the iron models, another model has been proposed. I call it the burnt toast model. And it works like this. Oxidation products of glucose, carbohydrates, particularly glucose and lipids, will form a degradation resistant layer around the tissue. They will also, like the iron model, they'll also work to increase the cross-linking occurring, and they'll also give a brown color to the tissue, because your toast, especially your burnt toast, but your toast has a brown color. It's because of these oxidation products. That's where the color comes from. So here's how it would work is we'll say the squiggly lines are the tissue. And then we have these oxidative products then coating that tissue such that they create a barrier that water can't get in, which also then means that water can't bring the solubilized iron in. So this is cost purposes with the iron model, because you're not getting the iron in to preserve the tissue because it's got this barrier keeping the water out. That also means then you're keeping the microbes out as well, which would enhance preservation. Okay, so that's what this model proposes then, is essentially you're coating the tissue with a layer that doesn't allow anything else to come in, including iron, apparently. All right, now, some of the challenges to this model. Okay, not all the dinosaur tissue that's been extracted is brown. Some of it is white. And that doesn't lend itself to this model because this model, its presence is detected by the brown coloring. Also, as I said before, cross-linking reduces elasticity and the changes to the chemistry that would be necessary to occur. And this actually applies to the iron model as well. You're going to change the chemistry of the amino acids when you're causing all the cross-linking. And yet we're finding intact, not altered amino acids. That's how we're able to identify the proteins. We're not identifying the proteins based on the alteration of amino acids. We're identifying the proteins based on the presence of the amino acids as they naturally exist. And I think one of the big problems of this model, of this preservation model, is it lacks any experimental verification. The paper that presented it just simply says, this is just what we speculate. In essence, they didn't use the word, but they should have. We're just speculating this is what it, so it's pure speculation. It doesn't have a single bit of experimental verification that this model would actually work to provide the level of preservation necessary. So what they in essence are proposing is that this model would help to preserve dinosaur tissue as long, if not longer, than synthetic polymers. In other words, we're wanting this dinosaur tissue to last as long as plastic. And yet we're not making plastic out of it, but we want it to last as long as plastic. Then I want to end with something I think has gone totally under the radar. Jeffrey Botta, who's a geochemist at the Scripps Institute, or at least he was at the time of this quote, when he first heard about Schweitzer's work back in 2006, he discounted it. He said, I cannot imagine soft tissue surviving billions of years. And here's his reason why. He said, even if the T-Rex had died in a colder climate, see, in other words, also recognizing that by their standard dating process, Hell Creek at that time would have been much warmer, okay? He says, even if it had died in a colder, drier climate than Hell Creek, environmental radiation would have degraded its body. Bones absorb uranium and thorium like crazy. You've got an internal dose that will wipe out biomolecules. And in fact, people that work with fossils, you recognize, you put a Geiger counter up to a fossil, it goes off. Fossils are radioactive because they absorb the uranium and thorium from the environment. Now, I haven't heard anything from Jeffrey Botta on this topic since 2006, but this still very much applies. The radiation didn't go away. If you're wanting to stick these bones in the ground for 100 million years and say somehow the radiation in the ground is not degrading the tissue, so you don't have any leg, you don't have any scientific basis to stand on. And so it really comes back to it, regardless of whether you are a big subscriber to the iron model or the burnt toast model or some other model, there is no preservation model that addresses the impact of radiation on these bones. So we close then with dinosaur tissue proteins are not consistent with the signed ages of the fossils, but it is consistent with the biblical creation model, which would say that these fossils were buried as part of the burial process of the Genesis flood, which was just a few thousand years ago, not tens of millions of years ago. So with that, we close. I have written a book called Echoes of Jurassic. There's also a video that's, it's not exactly a spin-off of the book, obviously, but there's a video where we talk about this topic. And then, of course, we have the special report, which is much more technical. And these can be purchased on the creationresearch.org website. So with that, I will close. Thank you very much.
Thank you, Dr. Kevin Anderson. What a great talk. I appreciate all the the different aspects of going through the details. You've mentioned what the claims of the evolutionists would be, how I think that's not supported. And yeah, just really having to force people to own up, I guess, to the discovery. And hopefully we see more. I think can't expect anything but more of that. Let's see if I can put us, trying to put us in our standard view. I don't know if you're still sharing. You can end your screen share.
I ended the sharing, yes.
Yeah. Thank you. And then, so yeah, we'll take some time. Feel free to add your comments in the Facebook Zoom or the Zoom Q&A, and we'll get through it. I think we can maybe even get in. I think one of the things that kind of stands out and is left open is really how the evolutionists will, their fallback, I guess, is the dating of the rock in which the dinosaur bones are found. Of course, they're not probably willing to actually date the bones themselves. There's one question, why do evolutionists dismiss the fact that carbon-14 has been found in dinosaur fossils, and maybe explain significance that type of dating would have?
The half-life of carbon-14 decay is about 5,700 years. So clearly there should not be any carbon-14 left in a specimen after, well, even just 1 million years, even just 200,000 years. But if you're looking at something you're claiming is 60, 70, 100 million years old, there should have, the carbon-14 should have decayed down to zero a long time ago. And so when we find carbon-14 still present in these fossils, then that to us is indication the fossils aren't nearly that old. In other words, they're still within the time frame of the decay rate of carbon-14. Now, the evolutionist community obviously cannot abide by that because that would contradict their argument. So they dismiss it like, well, it's contamination. Well, that's really nothing but a hand wave without really applying any intellectual activity into it, because contamination, first off, doesn't lend itself to equal. You know, if it's contamination, that means there'll be some here, not any there, some here, not any there. And that'll be, so it's not going to be a matter of that you're going to consistently find it, and you're going to consistently find it at about the same level. And yet that's what we find, is that when we analyze fossils for carbon-14, they are relatively similar in the level of carbon-14. And that wouldn't be contamination. Also, when you're saying contamination, you're just grasping at straws, because what is your method of contamination and where's that coming from? In other words, we're just assuming contamination because we don't have any explanation otherwise for the carbon-14. So they throw that out there without any really basis of saying that it's contamination, other than it doesn't fit their model. That becomes their biggest evidence that it's contamination. It doesn't fit their model.
Yeah, but to me, what was most perplexing is, are they saying that the lab, like in particular, finding that sequence of ostrich bone collagen, like, had that lab just been dating ostriches, or had ever dated
right, and that becomes, and that's something like that's been posed several times, that, well, there was contamination in the system. Mm-hmm. Well, the contamination in the system's been there for how long? Are they that dirty? You know, a good analytical lab will clean its stuff very well. And so in a sense, you're insulting them by saying, well, we're so dirty, we can't keep our equipment clean. You know, that's what you're in effect saying. But see, again, the desperation of trying to suggest that somehow this isn't actual dinosaur tissue.
Well, I think like the lab could at least they're stuck in a quandary. Like they could say, oh, we did data ostrich bone, but I think how likely would that have been? Like they can actually present some evidence that it is contamination, but they won't present the evidence to back that up, I guess. Another question here related, maybe we could stick on the like with Rana's paper saying that radiometric dating methods are reliable. This person says, but all radiometric dating make 3 assumptions. 1, no decay product was present initially, or that initial quantities can be accurately estimated. The decay system was so close to outside influences throughout the years. And 3, the decay rate was constant over time. Wouldn't a worldwide flood violate these 3 assumptions?
And of course, the answer to the last question is absolutely, and violate those assumptions. But what's amusing is that no knowledgeable geochemist will say that the system's entirely closed, or that there was no daughter in the system to begin with. That's been demonstrated time and time again to be the case. The system isn't entirely closed, and you do have daughter. In fact, what's called parentless argon in the potassium argon dating method, you have argon potentially present in the sample at the very beginning. It's known as parentless argon. Dr. Rana in his book even talks about parentless argon. But it's like we discount it because we know the specimens are so old that parentless argon won't be that big a deal. So they discount it because by their interpretation, it's meaningless. But by our interpretation, it's very meaningless. In other words, if the fossils or the sample, in case of argon, it'd be the lava, is actually young, then parentless argon is going to be very significant. So the idea that we're assuming that the systems are closed and there is no daughter to begin with, but yet we know that's not the case. They've already contradicted what is known to be the situation in order to get the date that they want. And I'd say one other thing about radiometric dating, and I think this is very important to understand. The relative ages of geologic formations, the relative age of the Rocky Mountains, the relative age of Grand Canyon, the relative age of the ocean basin, the relative age of these was already established in the literature before they even knew anything about radioactivity. But we also didn't have the analytical ability of working with radiometric dating methods until the 1950s. So you have to understand that it was 100 years of Darwin and Lyell pounding on the idea that everything is old, all these changes occur slowly, everything's got to take a long time before they even came up with the radiometric methods, which are now claimed to demonstrate the great ages. And I would challenge that the radiometric methods are a product of the assumption of the great ages, not a objective external verification. Had they used the uranium lead method, for example, and it came up with the date of 1 million years on what they had already predated at 50 million years, the conclusion would have been, well, obviously this method doesn't work. I promise you that. It would have been, well, this method doesn't work. So instead of it being the objective method verifying the date, it is a product of the assumptions of the date to begin with.
Yeah, not only that, I think you look at how, I guess, that uniformitarian assumptions, like we were talking about, that are undergoing sort of that it has to be millions of years old. Like, I think there aren't the same processes happening today that happened in the past. I think we could point to many things: volcanic eruptions, asteroid impact, which is a question here. So how are you going to lay down, like, people who studied flood depositions, the tsunami, storm, hurricane, whatever, the most they can lay down in sediment is like 10 centimeters. How are you going to cover a whole dinosaur fossil with an event that's can only ever lay down, that we've observed can only lay down like 10 centimeters? So apparently their whole system of using the current rates at the current intensities to extrapolate backwards is doesn't fit observation.
And we have to recognize as well that Lyell did not, in his principles of geology, he did not bring forth a lot of data. In other words, they had sedimentation data based upon seashores and those types of things, which even Dunbar in his historical geology textbook says you can't really accurately use those. That's not very accurate to use. They didn't have much data to determine that this takes a long time for these layers to form. It was just speculation. That's really all it was.
Yeah, and even instances of kind of fraud, or like the Niagara Gorge, where the Niagara Falls goes, and Lyell dismissing eyewitness evidence of the rate of retreat.
Because that didn't fit the paradigm he was trying to present.
Right. Yeah. Question here related, I guess, how would the impact of an asteroid affect the timeline?
I guess it depends on what asteroid you're talking about. Are you talking about the asteroids such as what the evolutionists proposed that wiped out the dinosaurs?
Right, that's what I think. The question by Chicxulub.
And I'm not a geologist. It's interesting that I've been sometimes referred to as a vocational paleontologist simply because I've gotten into this, but I've gotten into this because of the biology, not the geology. You'd have to talk to people like Mike Ord and Tim Clary more about potential asteroid impacts. Certainly some creationists have floated the idea that an asteroid or certain asteroid activity was part of the flood. And where that takes them, then you'd have to talk to them about that. But it comes back again, the biblical creation interpretation is that there was a massive worldwide flood. And even if the Earth's history was tens of millions of years old, with evolution occurring all the time, as is proposed in the standard classic textbooks, the flood would have wiped out the record of all of that and read that on its own record. So as creationists that take the Bible as the word of God, the inspired word of God, we recognize then that the history that we're seeing is a result of that worldwide flood, not the result of tens of millions of years of decay and destruction and such.
Right. There's a question here I think is good. Often they radiometric test lava. Is that not ideal conditions for contamination?
Well, that's the potassium argon method, for example, is in the lava. And that's where I talked about the parentless argon. The assumption is that the lava is so hot when it's flowing, the argon will escape. And so when the lava cools, there'll be virtually no argon left. So all argon that's present then later is a result of radioactive decay. But that's been demonstrated to not be true. The lava will cool, still trapping argon in it. So it's known as parentless argon. Well, how do you know how much parentless argon you have? Because there's no way parentless argon doesn't have an orphan sign on it, saying I am an orphan, I didn't come from radioactive decay. So how do you know what argon you have in the lava that's parentless and what came from decay? And the answer is you don't. And when I was reading Rana's book, I was absolutely dismayed that he seemed to not be bothered by that. How do you know that all the argon you're measuring for the most part came from the parentless argon? Well, the answer is you don't know. It's just the way that the evolutionists would know is that it fits the date we want. Therefore, this is the level of argon that came from radioactive decay. One of the challenges I would have to Dr. Rana is he is identifying himself as a creationist, but he accepts the standard geologic timescale. The standard geologic timescale is based primarily upon evolutionary arguments, evolutionary interpretations of the fossil record, evolutionary interpretations of geologic periods. So if you're a creationist, why would you adhere to a dating scheme that is so dependent upon that which you claim you don't agree with? So they can't get away from the evolution interpretation in the timescale by trying to claim that as creationists, we don't accept that because they're accepting the timescale. So they're accepting the evolutionary interpretation.
Yeah, which use like where the fossils and evolutionary tree of life, wherever they find those.
Biostratigraphy is a big component of dating the fossil record. Biostratigraphy is 100% dependent upon evolutionary interpretation. We know this creature evolved before this creature, therefore this fossil is older than this fossil, therefore these rocks are older than these rocks. That's biostratigraphy, just down to a T. If you're non-evolutionist, why would you accept biostratigraphy?
Yeah, I guess the question here is, have you ever corresponded with Mary Schweitzer?
I have not. I have had friends that have corresponded with her. We tried to get her into our Echoes of Jurassic movie. And I don't want to speak critical of her because I understand the position she's in, but she's put herself in this position herself. She was actually strongly considering being in our movie, but then she talked to Jack Horner, her former major professor, and he pretty much told her how terrible and awful creationists are and don't have anything to do with them. And as such, then she was negative to being in our movie to the point that she got very rude with our production company. Now, we weren't the ones treating her bad. Somebody may have treated Jack Horner bad, and that happens. That's unfortunate. That if a creationist treated Jack Horner bad, I apologize for that because that was unnecessary. But we weren't the one doing that. That wasn't us. We didn't do that. And so she didn't need to react so negatively. So no, I've not reached out to her because she knows who I am. And she initially liked the paper that Mark Armitage and I wrote until Mark, of course, filed a lawsuit against his university. And then all of a sudden she didn't like our paper. And I thought that was a little cheesy because you liked it until you found out we were creationists and now you don't like it. So I've not reached out to her because I really didn't feel that it would accomplish much. And I might be wrong. Maybe I'm judging too harshly. Maybe I should reach out to her.
Yeah, I had read an article in Smithsonian, I think it was, where she mentions that she has this view that she separates her faith from science. So there's this.
Yeah, which is silly because first off, you never separate your worldview from your interpretation. And that's all science is. People, just for me to preach for here 10 seconds, people talk about, I believe in science, which is okay. I thought science didn't have faith, but they also say, well, science says, X, Y, Z. No, it doesn't. Science doesn't say anything. Science is a tool that provides us data that we have to interpret. And when you interpret, you're bringing all of your knowledge and understanding of the world into that interpretation, and that includes your worldview. And so when they say I separate my quote religion, and of course I would argue Christianity is not a religion, it's a relationship. But when you quote separate your religion from your science, what you're really telling me is that you're not being very objective because you're trying to, in essence, draw interpretation that you think is the correct one, not based on what you're thinking the world is, but based on what someone else is telling you the world is. So my Christianity is a very important part of my interpretation of my science.
Yeah, otherwise I think people may not realize falling into some Kantian philosophical distinction of,
Well, if I'm going to hold true to my conviction that we were created, then my interpretation will fit in that criteria. If it doesn't, then what am I doing? I'm not really saying that we were created. See, atheists are considered to be objective, which of course they're not. They're anything but objective. They're bringing with them the we weren't created perspective, and their interpretation is all based on we're not created. And we can get into the whole origin of life situation, which becomes amusing because origin of life, what that tells me is I don't have any scientific explanation other than God created, because that's all we know scientifically. The law of biogenesis, life comes from life. That's a law, that's a scientific law. And if I discount that because that doesn't fit in my paradigm, I have to have life somehow evolve itself, that's not scientific. That is me introducing a worldview that doesn't fit consistently with the science. So yeah, we claim objectivity, we really don't have. And certainly people can point out my lack of objectivity on some things, and sometimes it's like, yeah, you're right, I wasn't very objective on that. But a lot of times it's, no, you're just imposing your interpretation on my interpretation. That doesn't make my interpretation wrong, doesn't make your interpretation right. See, I have my interpretation, you have your interpretation. Let's look deeper at the data and see which interpretation seems to be more consistent with other data.
A question here about, you mentioned that cross-linking would make the tissue less elastic, but since we find the tissue still to be elastic, this is evidence that no cross-linking has occurred. Are there other ways to show that cross-linking didn't happen? I guess, are there other reasons to say that cross-linking?
Well, what I had mentioned was the chemistry of the protein. Yeah, if you had the reactions occurring in the tissue, within the collagen, for example, that would be causing so much cross-linking, it would be altering the protein structure, particularly the amino acid structure. That's just part of the nature of the fitting reactions. And yet we're finding these fragments of proteins where the amino acids are still intact. That is indication that there has been no cross-linking occurring in that region, and there's been no fitting reactions occurring in that region of the protein. Now that itself doesn't disprove the iron model, but it's not consistent with the iron model.
It is becoming more common for scientists to check for dino tissues now than in the past. If they can't,
absolutely, yes.
Yeah, especially if they can't make a good reason. Do you think they're going to stop looking for it because they don't have a good reason, or they just resort to these bad reasons?
Well, that's a good question, because in part that brings in the idea of, do we stop looking for something simply because it doesn't fit the paradigm we want? And we certainly see that some scientists do that. We certainly see that happening some. But I think, at least in the world at the moment, 50 years from now, I don't know, but the world at the moment, I think we at least have enough scientists who aren't so concerned about what impact this has on evolution, what the impact this has on, origin models, that they would continue to search and publish. So that's my speculation anyway.
I think so.
Certainly, I think that lack of a method of preservation doesn't dissuade them at the moment, because they're not worried about that. We know it's old, it somehow survived, we're not worried about how it survived, we're just finding it. And so that's kind of the mentality at the moment anyway.
Here's a question. Is there a range of dates where bones exhibiting what has been found could exist? Maybe a few years, up to 1 million years, I guess. What's the minimum, maximum, I guess, based on certain assumptions, maybe the best case scenario?
Well, as the data I was giving for collagen, the best case scenario is that you might find collagen still remaining in a fossil after 3.5 million years. You're really pushing it to say there's anything that suggests it would be 4 million years, and that's still really being very generous. Most studies suggest that collagen would not last but more than 1 million or so years, and again, that's under fairly pristine conditions. When you take into account the non-pristine conditions of the state of the fossil, you're looking at having to admit that maybe 100, 200,000 years tops. And again, that's not saying it's that old, it's just saying that's your maximum age it could be, yes.
So I guess, has anybody looked at taking some realistic temperature like profiles of where the fossils found going back in time? Like, could they use that to date it it might not be all that accurate, but like at least would it be hundreds of years out? Like what if somebody has looked at kind of doing some actual observational, using some realistic values, what would they come up with?
That comes back again to the whole thing with thermokinetic studies. The reason thermokinetic studies use the higher temperatures is because some of these proteins, particularly collagen, won't degrade very fast. I mean, it's a tough protein. It won't degrade very fast. And so if you're looking at, okay, let's measure its real-time rate degradation, bone collagen, real-time rate degradation at 10 degrees centigrade, you're probably looking at an experiment that will take several hundred years. And most scientists aren't interested in experiments that take several hundred years. You know, it just doesn't work. So that's why they have to do the higher temperature and extrapolate. Now, we have just completed the first step of what I would claim is probably the most prolific or most broad bone collagen degradation study. We have used a wide variety of temperatures, and it took us well over a year to collect the samples. Now, we haven't analyzed it yet, so we're still far from reporting on it, but we're using this to address the idea of, okay, if we're using 80 degrees and applying it to 10 degrees, does the math break down? Biochemists say, no, it doesn't break down, but we're gonna address that head on and say, let's see. Now, we went down to 60 degrees. That's the lowest, because anything 55, 50, you're talking now, not over a year, you're talking probably over 10 years. Because it goes up by magnitudes as you drop down every 10 degrees. But we went down to 60 degrees. And so we're going to measure several points along the line from 90 degrees to 60 degrees to demonstrate how well it still correlates, as far as you're not getting the dramatic degradation rate differences at high degree versus lower degree that Dr. Rana speculates that you would get, because bone collagen is not that unstable. Right. But also, and again, I'd remind, not just collagen, they found other molecules other proteins such as actin and tropomyosin and myosin, much less stable than collagen. Yeah. And you got to account for those as well. Now, nobody does thermokinetic studies on those for the simple reason that they're not archaeologically important. See, most thermokinetic studies were done because of archaeology. You're studying the Egyptian mummy, or you're studying the Iceman or whatever like that. And okay, let's look at just how long it takes for certain tissues in the body to degrade. And so they've come up with estimates then based upon these thermokinetic studies. Actin and myosin and such degrade so quickly, they're not worrying about those in archaeological samples.
Expecting to find any.
Yeah, and yet here they are in dinosaur bones.
Right. Yeah. And then I guess related, has there even been like DNA found?
I pulled those slides out because I knew I was going to be getting a little long.
Yeah.
Some creationists are going ahead and saying, yes, I still hedge my bet. Here's the situation. Mary Schweitzer and her team have taken several stains, but the primary stain they used is a stain called DAPI, D-A-P-I. DAPI is a very specific binding pattern to what's a minor groove in double-stranded DNA. I think it's the minor, not the major. Anyway, a groove in double-stranded DNA. It only binds to that. It requires that to bind. And when they stain these cells with DAPI, it lights up an interior segment of the cell in the area you would expect the nucleus to be, which was the DNA would be, which not only says you're not just inadvertently randomly staining the cell, you're staining a very specific area of the cell where the DNA would be. And the DAPI specificity to double-stranded DNA means you have double-stranded DNA in that. Now, nobody's extracted it and sequenced it yet, so I'm not yet willing to say it's dinosaur DNA. I call it tantalizing evidence of the presence of DNA. But then that brings up another problem for the iron model, because double-stranded DNA is already highly cross-linked. If you're going to impose fit and chemistry on it, you're going to disrupt that double-stranded cross-linking for a different kind of cross-linking, which means you're breaking up the grooves of the DNA, and DAPI would never stain. So if you have DAPI staining to the groove of the double-stranded DNA, there's been no cross-linking induced by iron.
So it looks like a lot of research available for Christians to get into, and lots of.
And that's why the Creation Research Society is in the work. Yes.
Yeah. question here. It's broken up. Charles Dickens wrote about unfossilized ink in a cuttlefish that was discovered by Mary Anning, mid 1900s. I guess we were talking about going back to earlier discoveries. I guess I don't know if the ink and cuttlefish. Have you heard of that? I guess I heard even I don't know if it was Michael Horner would agree. Like you go to these dig sites and you smell rotting.
Yes.
Like
it's been known for years by paleontologists that fossils stink. Yeah. Now, the assumption always was, well, it's the microbial biofilm around the fossil that's causing the odor. But yeah, the fact that fossils stink is certainly an indication that there's rotting going on.
Yeah. Because going back, is it possible to document from the fossil record the series of transitional forms that led up to any dinosaur species?
Well, again, I'm probably not the best person to ask this because I'm not a paleontologist, but I would say that as I read the textbooks, and I can't really go into specifics so much, but as I read the textbooks, most phylum appear and disappear unchanged. In other words, it's there and then it's not there. Yes, in your imagination, you might be able to work some sequence where this fossil, this organism evolved into this organism, but that's it. It's just in, I can take, and I have this in another presentation, I take the heads of alligators and I line them up and show this nice transition of evolution of the head of an alligator, except they're all contemporary. They didn't evolve into each other, but you can do that with bone structure. So when we're interpreting the fossil record, see, it's very easy to misinterpret by creating a transition, creating a lineage that really didn't exist. So, but whether there is what evolution identify as, well, this is the forerunner of this one, I'm not as familiar with that because I'm not a paleontologist. I'm sure they have some, but once again, the warning, in my opinion, is you have to be very careful. How do you know that's a lineage? Because we can do that with living creatures today, and it's not a lineage. It's just simply they have enough structural similarity that you can create depending on what you look at. Does it get shorter? Did it get taller? Did it get longer? Did it get wider? And so whatever criteria you're using, you can set up those kind of lineages using contemporary animals.
Yeah, or even they've had so much difficulty saying what species of dinosaurs are. Could later find out it's a juvenile or female or
exactly.
Like.
And that brings up another issue, and that is fossil species. Mm-hmm. And some paleontologists get really upset about this, and I kind of understand. But on the other hand, they need to, kind of put up their big boy pants and move forward. We can barely identify species of living animals. How are you supposed to look at a bone and say what species that is? See, you can say, okay, it's in this group, but how do you know that this bone and this bone represent two different species? Like you say, as you allude to, they then later find out, oh, this is the male and the female, or this is the juvenile, and this is an old adult. See, so they've embarrassed themselves several times by trying to too precisely delineate species within the fossil record where they weren't different species.
Well, they probably want to be famous for some discovery, right?
Well, you don't publish on identifying a species already been identified. Yeah. There's some 1,200 different species of fruit flies in the Hawaiian Islands. Are there really? No. It's just that gives me 1,200 different publications. A new species. See, so, in the field of classification, there is what we call the lumpers and the splitters. And it's more philosophical than it is, biological. If you're a lumper, then there's only one species. If you're a splitter, everything's a different species.
Mm-hmm. Question here, but does evolution explain the Cambrian explosion of every major animal body plan in a single rock system? According to evolutionary age assignments, this profusion of forms occurred in the lower Cambrian. Stephen Jay Gould, who is an evolutionist, by the way, for those who don't know, an elegant quote by him, or writes, an elegant study published in 1993 clearly restricts this period of phyletic, this I guess classification order, flowering to a mere 5 million years. Was this enough time for evolution to perform all that invention, all major body plans essentially within 5 million years?
Yeah, again, I'm not a paleontologist, keep saying that, but yeah, the Discovery Institute, in fact, in Seattle has done some really nice study, really nice publications directly focused at the Cambrian explosion and discussing and analyzing the arguments that the evolutionary community has given for the sudden appearance of all these phyla at that period of time. just suddenly appearing with no apparent history behind them, and moving forward in many ways relatively unchanged. And so, yeah, they've done, Discovery Institute's done some very nice work on that.
Yeah, I think we got a DVD of theirs on that. I think Darwin's Dilemma, I think it's called.
Yes.
Here's a question. I am assuming some fossils do not have any tissue present. Is it reasonable that all tissue could be degraded in a few thousand years? I guess of those fossils that they wouldn't have found. Yeah.
Yes, not all fossil has tissue. That is correct. And even as a biblical creationist, I would say that there are certain conditions necessary for you to expect preservation of the tissue even for a few thousand years. So yes, that's why you don't pick up any fossil at any place and suddenly say there's tissue in this. But clearly it's not just one location and not just one type of fossil. As I stated during my talk, number of different labs with a number of different specimens that are found all across the world have isolated tissue. So it is a widespread event.
So maybe this will be a wrap-up question here. Speaking of cancel culture, do you know of even one creationary scientist who has ever been interviewed on a major newspaper, TV, or radio, like NPR's Science Friday, CBC Radio Science Show, Quirks and Quarks, etc.?
We used to be. You used to see a creationist interviewed on, Fox, or interviewed on CNN or something. You used to see creationists were allowed to write comments. And I remember back, oh gosh, this has probably been 40 years ago now, when Duane Gish was able to do a written debate against Isaac Asimov in the pages of Science News, I believe it was. Science magazine back in the early 80s ran a feature article on creationists. See, so there was a time that I guess we were just kind of considered the crazy uncle, but they weren't too worried about us. But the problem is we've hung around and we keep getting more and more vocal, and we keep getting harder to argue down because they don't have arguments. They can't, what can they say? So now we've moved to that phase where ignoring is the way. We can't allow them to the table because what they say we can't necessarily refute, so they're just not invited anymore. Recently, certainly in more recent times, yes, you're not going to see creationists given that kind of platform.
Exactly, that's what they'll say. We don't want to give them a platform because I don't know if it was Dawkins who had some debate did so poorly. It's like, I'm not even gonna give him that's his excuse.
Well, part of it is he did so poorly. Yeah, exactly. So his excuse is, well, don't even give him the platform. But it really came down to he just did poorly.
So I think I don't know if we want to get into like the rocks that they're found, like wicking away bacteria. Like some people might say, oh, you can get reactive enzymes away because of that wouldn't degrade the bone because of the sandstone, anything like that. But,
well, that is true to a little extent, but you have to recognize that sandstone is still loaded with bacteria. In fact, there are bacteria that go thousands of feet down. I used to work for a biofuel company that worked with natural gas. And you say, well, I was a microbiologist. In fact, this biofuel company hired a lot of microbiologists because natural gas is a microbial phenomenon. It's not a geologic phenomenon. And it was amusing to me to try to get the geologists to understand that the gas that you're pulling out of the ground is probably younger than you are. Because they were taught that it's 50 million years old. No, it's biological and it's probably been made recently. What? That's 2,000 feet down. Exactly, 2,000 feet down, there are microbes in that very, very hot environment that are making methane, and that just blew them away. So sandstone is loaded with bacteria, and these fossils are not 2,000 feet down. They're just a few feet down on average. So those are a lot of bacteria, and of course that gets a whole bacteria biofilm argument, which I didn't go into because to me it's such utter nonsense.
Yeah, maybe this one for a very last question then. In the Jurassic Park movie, they made a reference to amber as a place where DNA was extracted. Comment on amber as a place where usable tissue could exist.
Well, first off, if you remember in the more recent Jurassic World movies, they've replaced amber with iron preservation of tissue. Yeah, which amused me. So we don't need amber anymore. But interestingly enough, there has been some work done on amber preservation of DNA, and it doesn't work. It doesn't preserve the DNA.
Yeah, because I guess they say they can withstand high temperatures or something.
They can withstand some. It certainly gives a preservation, but it doesn't give a preservation near long enough to be able to get DNA out of some kind of sample that supposedly is, 60, 70, 80 million years old. In fact, I think what it was is they couldn't even get DNA out of samples that were, just a few thousand years old. In other words, amber just simply did not work as a preservative.
Interesting. Great. Well, thanks for enlightening us. I think lots of valuable information. It's very much, I think, strengthens our faith in understanding what God plainly said. Like if just taking God's word straightforwardly, we can go back and the origins account that God's given us is trustworthy. I want to share with people our website. If you want to get more information, please go on to creationbc.org so we can show. We plan to have a speaker come up at the end of May. We want to solidify the dates first. Dr. Jerry Berman, who, again, another Ph.D., a scientist. You can look on our events page here. We also have, in case you want to be put on our mailing list to hear when these next events will be, check us out under the Contact Us section, so About and Contact Us, and fill out this form. I promise not to spam you. I want to also alert you to all the articles. We have many articles that this is just a starting point, I guess. Just to immerse yourself, be informed. Again, if you're being challenged, be prepared. Do a search if you're into dinosaurs. We got on our books, our online resource store. Click the store icon, and then within there you have a search bar. Separate from this search, I should just quickly mention. Search for dinosaur. You'll get up many things like for kids, an activity book, or many like evolutionists might use, oh, you got dinosaur tracks at different layers. How could that be? But Mike Ord's done great investigation research into questions like that. Buddy Davis, a video kids would love, very highly animated. Many other ones. So Prue's there from Institute for Creation Research. A good one by Tim Clary. Again, if you liked this talk, want to share with others in the DVD format, including yesterday's, Dr. Kevin Anderson features in this is Genesis History, Volume 2. It goes into interviewing a number of PhD scientists on biology, each in their own respective experts in their field. Also, highly encourage the actual movie is Genesis History. Put on by Decay. For kids, or many questions get brought up by kids. So we got this collection of 8 volumes, each one dealing with questions that are often asked by kids. Each one is $8. If you just want to get one, you can look at hopefully the table of contents in there. And then just again, if you're kind of being introduced to this, you want to get an overview. There's this guide to creation basics with nice colorful graphics made for the older youth, but also there's a more wordy one. I think it's similarly titled something like Guide to Creation by the Institute for Creation Research that goes into the sort of, if you want a primer to learn about this some more, get foundationally informed. It doesn't take a whole lot, I don't think, to just be relatively informed to help you understand when you're brought up, you hear some evidence interpreted from an evolutionary view, that you can be informed and have a valid interpretation that starts with intelligent design or the God of the Bible that will make sense of the same evidence. Because it's how we interpret it. And I find usually the creation looking from the biblical view makes much more sense and puts all the pieces together, I think, more coherently. Sure, there's still problems, but that's areas for research. So I want to thank again Dr. Kevin Anderson. If there are any other closing remarks, we'll thank everybody for joining us.
Thank you for having me.